So this is a pretty typical of this class that we are a little bit behind. So we're going to finish up lab 1. So hopefully everybody ended up running multi-QC and has a multi-QC file. So let's start. Let me make this really big. Everybody can see it. So we are going to log into Beacon. I'm going to SSH MC1 beacon.nix.utk.edu. And I have my car keys instead of my office keys. Not going to be useful for logging in. And we are going to, actually we can probably do this from the login node for right now, because as soon as we get done with this, we're going to move into a lecture. So from here, I'm going to CD to Luster Medusa, and then your student name. At this point, you should be able to see your RNA-seq directory. And if you follow along with our file system, we're going to go RNA-seq analysis 03 multi-QC and do ls-l. So we've got a lot of stuff in here, which is really good. But most importantly, you should have a multi-QC report, that HTML, and it should have some stuff in it. So it shouldn't say 0 right there. So if you are not able to find this file or you didn't get to this point, throw up a red sticky, someone from the back row can come help you. Okay, so file transfers. Oftentimes you're gonna need to move data between your laptop and Beacon, or in the future, the ACF, and get that data to go both up and down.
Just a question, those two files we linked yesterday, right? So once we linked it, it's always there.
Yeah, that symbolic link stays there forever until you delete it.
The deleting means RM.
Yes, and that if you use RM, late flower rep1 tinyfastq.trim.log, it will just delete the symbolic link. It will not delete the file that it's linked to. So yeah, those are persistent. They'll stay there. It's like its own little file pointer, and it lives in the file system. And when you log out, it still is there. And when you log back in, it's still there. Okay, so there's 2 ways to do file transfers. First is secure copy, which is very much like copy on the command line. So you have to deal with paths and specifying where something lives and where you want it to go on your own laptop. So that's a little bit more technical. The second way is FileZilla. FileZilla is more of a GUI. It can be very convenient if you're not as familiar with paths. However, with the token system, it's kind of a pain because you have to type in your ID from your token about 4 times to get anything transferred, which is difficult. So we're going to start with secure copy. Now, is there anyone in here who is using PuTTY on Windows? Okay, if you're using PuTTY, you do not have a secure copy function in PuTTY, so you will have to use FileZilla or install something like Movaxterm. So you can ignore this next little bit while I show the rest of the class how to do secure copy, and then we will do FileZilla. Or actually, to save time, do you want to have like a little FileZilla group in the back? Would that be cool? No? Okay, we'll see how secure copy goes, and depending on the number of people who don't get it to work, they can also join the FileZilla. So I'm going to start by just typing pwd so that I know exactly where the file I want is. And then I am going to open a second terminal, which is something if you're a command line person, if you end up doing a lot of command line, you're going to quickly realize that it can benefit you to have like 3 or 4 or 5 terminals open, all doing different things. So yes, you can have multiple terminals open to the same computer and be running different jobs all at once. That is very feasible. So I'm on an Apple, so Apple has its own very Linux-like operating system. And I know that just because I'm familiar with the operating system, if I want to move to the desktop, it's right inside of your home directory. So navigate to your desktop, users, whatever your username is, desktop. On Windows, it can vary a little bit based on your operating system, but it should be something like C/users/your username/desktop. So do pwd, do ls, and try to cd around until you get to your desktop. How many people are on their desktop in a local terminal? Okay, looks like I've got some yeses. Okay, awesome. From here, we're going to secure copy. So the command is scp. If you're on Mobaxterm, if you're on a terminal on Apple, you should have the secure copy command already. Let me see, mstate1 at beacon login. I don't know if it matters. Anyway, login.beacon.nix.utk.edu. So this looks exactly like our SSH command, except that we put scp. Now here's where the first change comes. Now we're going to specify the location of the file on the system. So we're going to put a colon, no spaces, and then type the full path. And I don't want to type this whole thing out. I'm going to copy and paste it. And then I'm going to put the name of the file. So one giant long string here. We got username at the remote location, so that's the server, colon, and then the full path to the file you want. And I'm going to say, I want you to go out into the internet and find this resource and put it right here. And that single dot means put it, I'm on my desktop, so put it on my desktop. Type yes. You will have to do your passcode again. Oh, no such file or directory. What did I type incorrectly? Multi-QC. Okay, so I forgot the Q and the C. You guys are supposed to be helping me out when you see me mistype things. Okay, so you know it works when you get this little 100%, tells you how much it copied, the speed at which it copied. So it's a very small file, so it should copy super fast. Okay, green sticky when you have your file copied over. Red sticky if you're having problems and somebody will come help you. No sticky if you just want to be left alone to mull over the whole thing and try to make it work by yourself. Yes, and if you are an enrolled student, this is the moment, email me the file. Get your 5 points. All right, I don't see any red stickies, so we're gonna move around. So this is great, this is on our desktop. And that means we can just double click on it and open it. Hopefully, if I can find where it went. There it goes. So it has created this lovely HTML output so that we can look at our files and see what's going on. Up here, you'll see this handy watch a tutorial video. If you click on that, it'll take you to YouTube. You can learn a whole bunch more about the multi-QC interface and all of the interesting stuff that you can find out about your data. The first thing at the top, it's going to give you what % of your data were trimmed by skewer, what % of your data were duplicates, so sequences that were identical. This is not something I worry about too much in RNA-seq data, just because you do have genes that are highly expressed, and so they eat up a lot of your sequencing. This is a bigger deal if you're doing DNA sequencing, because you have the same read that's being sequenced over and over and over, it could mean you have some PCR problems. It could mean you have some contamination. It's not something I worry about too terribly much in RNA-seq. Percent GC and millions of sequences. So we have 250,000 sequences, so it's a 0.2. It also gives you this read length distribution after trimming with skewer. So we started with all of our reads at 51 bases. So it would have been 100% at 51, but now we have a good many that are 50, 49, 48, 47. So not too much trimming going on. And so, okay, this is another thing. So multi-QC has mushed together your skewer results with your fast QC results. So you have general stats about your files, you have your skewer results, and then it gives you your fast QC results. So this is the sequence quality histogram. So you can see these reads were excellent. There was not a lot of trimming that needed to go on. The mean quality scores are all the way in the green. So this is starting at base pair 1, 2, 3, all the way out to the end, so base pair 50, 51. If you sequence 300 bases, this will go all the way out to 300. And in our case, we didn't see that drop off of quality, which is great. Usually the longer the read, the more you're gonna see that drop off in quality at the end. This is a similar metric, but looking at the average quality score across the length of the read. So this is the mean quality score by the count, the number of reads. So the vast majority of our reads have an overall average quality from 30 to 35, which is very good. So it's 95 to 99% accurate. Here we have the per base sequence content. This can be kind of hard to see on the projector, but you can, since we only have 2 samples, there's only 2 rows, but I'll show you an example in a minute where we have more. But if you click on one of your samples, it'll give you this nice little chart for the sample. And so this is so typical of Illumina data, this like chaos right here. And it's because when they ligate the primers onto the first part of the read, it's not truly random. They use random hexamer primers, but it's not random. So this is super typical. I don't ever worry about it. The reads map fine. It's real data. It's just not normally distributed. But it is usual. You would like to see for the rest of the read to be normal, kind of consistent. And we have, so if you did have a lot of, if you had 50 reads, you could click through. But we only have 2, so it's just going like file 1, file 2, file 1, file 2. You can go back to the overview heat map. So these 2 look very similar to each other. So another thing I'm looking for here is just any sample that stands out and looks really different from all of our other samples. Our GC content, pretty consistent between the 2 samples, nothing super wacky going on. We don't have really any ends, so we don't see a sample where lots of the bases were called as ends, which would indicate some major sequencing failure. Our length distribution, again, most of our reads are quite long. In comparison, I mean, we started at 51 bases. They have not been trimmed down to be like 25 bases or 20 bases long, so that's good. Our duplication levels, I suppose it's a little interesting that the early flowering has some larger set of like a spike in duplications. But in the scheme of things, this is gonna be less than 2% of the library, so not super worried about that. Again, duplication is something I worry about a lot more for DNA sequencing. We do see that our early flowering has some overrepresented sequences. So we might wanna go explore that. And no samples found with any adapter contamination. So fast QC will check for the most common Illumina adapters. If they have some crazy library prep at the sequencing facility or their own custom adapters, it will not find them, but it is going to check for the TrueSeq and NextHair and the absolutely most common ones. Okay, so this is overall a pretty boring multi-QC. But I asked Wenxing, she had some really cool chicken data that we were processing a few weeks ago, and I thought I transferred it onto the computer. Oh yeah, and we need to go do FileZilla, sorry. See, you guys are supposed to interrupt me and tell me, Meg, you're screwing this up. Go do FileZilla. Like seriously, do not be afraid to interrupt me. which we may not even have installed on this computer. Okay, so that's okay. We can do it from my computer. All right, sorry to anyone watching the video. You're not going to be able to see FileZilla. Hold on. Okay, so FileZilla looks like this. It's pretty nice if you appreciate GUIs more than command line. You're gonna see it's a little bit of a pain because you have to type in your PIN a million times. But how I do it is go to Site Manager and put in your information here. So you're gonna put in the login.beacon.nix.utk.edu. You can put in port 22. I think that's also the default port, so you may not have to. Importantly, you have to choose SFTP, so secure, SSH secure file transfer protocol, and you have to choose interactive. And the interactive is because of the token system. Because it changes every 30 seconds, it has to be interactive. And then put your username. And if you click connect, mine pops up a little box. Hopefully yours does too. And this is your same, like put your PIN and your 6-digit code. And then all of a sudden, so this pane over here is the file system on your laptop. And this pane over here is the file system on Beacon. So now you can navigate to your files on Beacon. And I'm just gonna change this up here to Luster Medusa. And then hopefully, once you'll be Luster Medusa in your student name, and then you should see RNA-seq, analysis. So this is so satisfying, right? Like we've been talking about visualizing your file system on Beacon like this, and then you finally get to do it. Like you can actually see the files that you've created and navigate them in like this normal way. And then you can go to multi-QC. And then over here, you wanna navigate to your desktop. So again, on a Windows machine, probably gonna be something like users, I don't know, I'm not very good with Windows. So for Mac, it's gonna be users, your username, and desktop. So once you've found that, you can just take your multi-QC report and drag and drop it. Now it's gonna ask you for your passcode again. So target file already exists because I've already transferred it. So I'm gonna cancel that, but hopefully yours will actually transfer. Is that working out for the FileZilla people?
So we can brand it now?
You can brand it?
Yeah, we can brand it. FileZilla.
Yeah, exactly. It's your own little group, the FileZilla people. Mm-hmm. We can have like Team Secure Copy and Team FileZilla duke it out over which one's better. All right, so while people are working on that, I am going to show you WinGene's data because I think this is exactly when you run FastQC and then multi-QC what you want to really see. So she has 32 samples, and so it's a lot more convenient. Instead of us trying to click through 32 FastQC files, and they're paired in, so it would actually be 64. It would be really a lot to look at all of them. Instead, it's going to conglomerate or aggregate the statistics so you can kind of do comparisons. And so we can see her data is also RNA-seq, and she has a much higher % of duplications. So she has a lot of reads, a lot of genes that are probably very highly expressed. So but I'm not worried about that. But it also has millions of sequences. So she can see that for some of her libraries, she got 3 million reads. Like this one in particular, she only got 3 million reads. For some of her libraries, she got 37 million reads. So a pretty big disparity in how deep these libraries got sequenced. So she could potentially, you know, if she felt like 3 million wasn't enough, she could call the sequencing facility and be like, I need to put, you know, maybe the IC2 library back on a sequencer and get some more reads.
For the percentage of duplicates, that's for the first column. The second column, what's the duplicate exactly?
What percentage of reads are highly duplicated?
Oh.
So it's a lot of times if you're like,
it's the same gene.
If you have a couple of genes that are really, really, really highly expressed, you're going to sample those genes a lot, and then those reads look like they're duplicates. In a DNA sequencing experiment, this would be an indication that you had usually not enough DNA going into the library prep, and your PCR created a lack of complexity in your library. But you can't really tell if that's what happened in RNA-seq, because you expect an RNA-seq to have genes that are expressed more than other genes. You hope in DNA that you've evenly sampled the genome, and so you shouldn't have a lot of duplicated, I mean, you'll see some because of transposable elements, but it's much, you see a lot higher percent duplicates in low complexity RNA libraries, just because if there's certain genes that are highly expressed.
How would
I don't think it would make that big a difference, because I think you would still have lots of different genes expressed. Your total sampling, I don't think it would make that big a difference. I think you would still see a significant percent of duplicates, but it's something that, like, I wish we had better ways to tell if it's a library quality problem or if there's like really highly expressed genes, but you can't really tell with RNA-seq, so I just ignore that column, essentially, for RNA. But here's where the power really is. So if you look at the FastQC files, you would only see a single line for each library, but MultiQC has aggregated these, and you can mouse over and see all of your libraries. And so this is a great sign. Her library quality is super tight. Like all 32 samples are all completely high quality and look really good. Same for the per sequence quality scores. We see a little bit of differences at the very last base, but that's it. So very consistent. Here you can see her heat map. I can actually see when I look at it, there's like this region right here where the color looks a little different. So if I click and we see kind of a standard, and then you can click next, next, next. We look super consistent, pretty consistent, pretty consistent, pretty consistent. And then eventually, okay, so that one looks a little different. Don't really know why. So that was my first indication. LT4 might have something weird going on. IC2 might have something weird going on. So anything that doesn't look like the others is usually a red flag. Per sequence GC content. So when I got to this, I was like, whoa, whoa, what happened here? So most of the samples seem pretty reasonable. We've got this one sample, IC2. IC2 is off on its own little GC peak. Like that seems very weird. If we're sequencing chickens and we have biological replicates, why would we have this? So this is when, as we were going through the data, I was like, okay, we really need to look closely at the sample. Per base N content, again, I'm seeing this one sample, IC2, has a significantly higher percentage of N's than any of our other samples. These two, LT3, IC3, you know, they're a little concerning as well, but IC2 is really sticking out on all of my graphs.
Is any of these samples the one that was like the very low number of reads, like 3 million reads something?
That is a good question. I can't actually remember. So we were looking IC2. Yeah, very low. So it seems to be biased. It doesn't have enough reads. We also looked and it was contaminated with, Jean will remember, What did we find in IC3 or IC2 that wasn't in the other samples?
IC2.
IC2, what was the, was it E. coli? What was it we found in there?
I think yeah, some bacteria.
Anyway, there was this bacteria contamination. The chicken RNA isn't showing up that well. It was low sampling, it was contaminated by bacteria. It was just a problematic sample from beginning to end. That's the type of thing you can see from your cross QC report. So for multi-QC, I would look for anything that doesn't look like everything else. Usually it's time to go start digging. Okay, so we're 11 minutes behind, but that's okay. I always think things are gonna go so much faster than they do. Did everyone get their files transferred? And okay. Okay, I'm gonna switch back to the other computer now so the recording will work out. It's almost the whole slide. Okay, so I'm going to give you a little bit of background about project design. And a lot of these questions came up yesterday, so we've covered some of this material, but I think it feels like this will be of interest to many of you. So the major considerations for project design, I would say, of course, cost sort of dominates everything. You only have so much money. If all of us were billionaires, it would make science a lot easier, but we aren't. And so, so you kind of have to balance your needs statistically with the amount of money you're going to spend. So the two things that you're thinking about when you're trying to take your precious dollars and divide them up into sequencing is how many reads do I need and what kind of statistical experimental design do I need as far as replicates. And at this point, caveat, I would say it's really good to talk to a statistician or a bioinformatician or both before you ever set up your experiment. I would say probably about once a year since I've gotten here, somebody has come to me with data and they have done something that makes the data completely confounded and unable to be analyzed. And then you're like, I am really sorry you just spent a bunch of money. I can't help you. Your data is useless. Please don't let that be you. Just try to talk to your statisticians up front. Make sure you have a good randomized block design. Make sure that you're getting your biological replicates. Make sure you're doing your libraries right. Because you do not want to find out after you've paid for your sequencing that it's not going to work out. So what do I mean when I say replicates? I keep saying replicates, and there's actually two types of replicates that we talk about in science. The first is biological replicates. So we want independent biological samples processed separately. And in the case of experiments, for sequencing experiments, we want them barcoded separately. So true independent biological replicates. There's another type of replicate called a technical replicate. This might be the same biological sample, but you've done independent library construction or independent, something in the lab you've done differently and produced two samples. This can be, technical replicates are really good for verifying experimental variation due to the sequencing machine, due to your library prep procedure. If that's what you're trying to quantify, then you do technical replicates. But for the most part, if you're asking questions about the biology, you want to do biological replicates. And the reason why technical reproducibility is extremely good for RNA-seq. It is much better than microarrays, but biological variation is huge. So you want to put all of your statistical power into detecting and understanding your biological variation. Do not waste your money on trying to understand technical variation because it's going to be so much smaller than your biological variability. So always do biological replicates and not technical replicates, unless your experiment is, I'm testing two different library preparation procedures, or I want to quantify the variation on this new Illumina sequencer that I just bought, in which case you would want to do technical replicates. So yeah, so understanding the difference between those two is very important. The question I get asked very frequently is replicates. How many do I have to do? And this does depend on your organism of interest. It depends on the journal that you're targeting. But there's excellent evidence that beyond a depth of 10 million reads, replicates provide a lot more statistical power than depth. So as many replicates as you can afford is probably the right answer. If you have one replication, it's gonna be really hard to publish. You may be able to do one control and one experimental next generation sequencing run and then go verify your gene expression with RT-PCR or something like that. But in general, 5 to 10 years ago, it was not hard to get published with one replicate. Now it's nearly impossible. I still see publications coming out with 3 replicates. I think that's changing quickly. This paper came out a couple of years ago. I think it was, they really went the distance to really test biological replicates. They went all the way up to 48. Which none of us I will never be able to afford. Okay, that's not true. Prices are coming down, but 48 replicates is a lot. But they really wanted to see what happens if you add replicates. And so with 3 biological replicates, 9 of the 11 tools found only 20 to 40% of the significantly differentially expressed genes. So you might very well be leaving 80 to 60% of your real data on the table. That would make interpreting your results difficult. So their suggestion is at least 6 biological replicates, rising to at least 12 when it's important to identify significantly differentially expressed genes for all fold changes. So if you're looking for tiny changes, you gotta go really deep. You gotta get a lot of biological replicates. And they also say if fewer than 12 replicates are used, you should consider statistically using EdgeR or DE-Seq2. So we've chosen in this class to teach DE-Seq2, and this is one of the primary reasons. If you're the type of researcher who can get more than 12 replicates, you're gonna want a different statistical package. But for the most part, I almost never see anyone who can afford that many biological replicates. So we're gonna stick with DE-Seq2, which is, it's good for low replicate experiments. And I should also say this is done in yeast. Yeast has a lot fewer genes than a lot of our organisms. So I imagine as you scale up to many, many, many tens of thousands more genes, the noise gets bigger, the variability gets bigger. It could very well be the case that if you ran this experiment in a plant or in a mammal, that they would come up with even more biological replicates. So that's the disappointing truth. So sometimes this means scaling your experiment down to have fewer treatments or less complexity so that you can add in the biological replicates that you need. To keep belaboring the point, this is a study that did a nice job with looking at the number of replicates. And so the number of differentially expressed genes, so this is really funny because I can tell these people didn't want to pay the color figure charges, which is why we have the weird, like super thick lines. I've totally been this person who has tried everything in my power to make pictures grayscale, so I did not have to pay color figure charges. But yeah, so as you increase the number of replicates, and so yeah, so let's just look right here. As you increase the number of replicates, you're really vastly increasing the number of differential genes that you're discovering. And if you compare that to adding depth of reads, you're not making as much progress as far as adding real differential expressed gene sensitivity. So in this case, you can literally go 6 times as much depth of sequencing, and you're not going to make as many gains as if you just added like 4 biological replicates. So replicates are giving you more bang for your buck. And the same thing over here with the power at FDR 0.05. So your power is going up vastly when you add replicates. And what's interesting is Jim was showing me some data from colleagues at a conference, and they were showing the opposite, which is the number of differentially expressed genes was going way down with biological replicates. And at first that may seem surprising, but I don't think it is. I think by adding biological replicates, you're controlling false negatives and false positives. So you're drilling down to the truth, and whether that means that you're discarding false positives, that is just as good as gaining more true positives, right? You want to get accurate. So I think statistically you're controlling both type 1 and type 2 error by adding biological replicates.
Just a quick question about this paper's results. So if I have limited budget, should I increase the biological replicates or increase try to get more reads? What's the result?
I would go more biological replicates,
Okay.
for sure. Yeah. So if you spent a whole bunch of money and got a whole bunch more reads, this would get you out to some number of differentially expressed genes. But if you add your biological replicates, you're much more quickly going to get to the truth. Even if you didn't do the additional sequencing per replicate, you would still do better. More differentially expressed genes would be identified than if you just did more depth of sequencing. That's how I would interpret that. And I put the citation so you can go find the paper. And there's a whole bunch more information in there that's really great. So many of these papers you will see, so there's lots of statistical packages that you can use to analyze your data. And I'll actually talk a lot more about this tomorrow or Friday. But most of these papers recommend EdgeR or DSeq. And they're both going to use your replicates to try to figure out the variability of expression of each gene. And this is a hard problem because some genes are more variable in expression than other genes. Some genes are under tighter control and are consistently expressed at a certain level. Other genes seem to be much more variable. So when you only have 3 or 4 biological replicates, it can be hard to understand, to model that variability. So both of these packages attempt to do that. Simon Anders is one of the authors of DESeq. He says, of course, we like to claim that DESeq is better than EdgeR, and for only 2 or 3 replicates, I do think so, but for 5 or more replicates, EdgeR's moderation feature really pays off. So this might be your tipping point if you move over into EdgeR instead of DESeq. And from the S Church paper, which was the 48 biological replicates, anything less than 12 replicates, they recommend either package. If they go over 12 replicates, they recommend DESeq. So this is actually, and DESeq2 and DESeq are different, and they do handle variability differently. So if you're kind of teetering on the cusp of having more biological replicates and you're wondering what package to use, I would read that S Church paper for sure. It's just, it's all in how they handle the variability and the statistics of the variability. And there's a lot of statistical manipulations that go on with low biological replicates, because you have to kind of make some assumptions that may or may not hold true when you actually add in real data. So. Okay, another thing that people like to do, pooling. Does pooling my samples count as biological replicates? My answer is no, it doesn't. You're gonna get an accurate mean, hopefully, if you've mixed your samples in relatively equal proportions, but not an accurate measure of variability. So that's the problem with pooling. The literature is very mixed on this issue. There are some good papers out there that say pooling is fine, but you're gonna talk to any statistician and they're gonna hate pooling. Statisticians think that pooling is the worst thing ever. They're just like, why would you do that? Because the downsides are significant. You don't have real data for each independent biological replicate. You cannot measure variability in the system. And there are some papers that talk about how it can really give you low positive predictive values. So you're going to be much less good at detecting differentially expressed genes. So I in general do not recommend this, although I would say it's probably still publishable. And my argument on that too, I shouldn't say, the fact that something is publishable, hopefully it's not the end all and be all of science. What you're hoping to do is detect actual biological reality. If you're gonna build your research program for the next 10, 15, 20 years on a result, you want that result to be accurate. You want it to be robust, you want it to stand up. So if that's really your goal, is to do solid science, don't pool. So going back, harkening back to those statistics classes that hopefully everybody has taken, you want to follow good statistical practices, randomized block designs, for example, just revisiting those briefly. So if you divide your samples or individuals into blocks in order to control variation between the blocks, and randomly assigning individuals to those blocks. So something like this might be, I have my early flowering cultivar, my late flowering cultivar. I'm going to randomly divide my 40 individuals between 2 locations. So that would be a blocking experiment, a randomized block design. But it turns out you don't want to just use that in your upfront experiment. You can actually may need to use that downstream to control some of the problems that can arise during sequencing. If you run samples across different lanes on a high-seq, that can create problems. You can get batch effects, you can get lane effects. So certain lanes may just produce higher quality data than other lanes, and if you put all of your treatments on one lane and all of your controls on a different lane, you have confounded the ability to detect that. And I didn't used to teach this, but there is this cautionary tale that came out, made me think I should mention this. There's an original paper doing the comparison of the transcriptional landscapes between human and mouse tissues, and they found that gene expression did not group by tissue. So if you look at the brain of the mouse and the brain of a human, there were no similar expression patterns. And it turned out they had sequenced all of their human samples at time point A, waited 6 months, and then sequenced all of their mouse samples, I think at a different sequencing facility. And so there were all these like really big intrinsic differences in the sequencing. And when some statisticians went back and pointed out this flawed statistical design and really questioned the results, they found the exact opposite result of what was published in this paper. And this was a PNAS paper. It was a very, high impact journal. And so this made Twitter blew up. This made huge news. So it's worth thinking about how to actually use randomized block design. And this is what is currently recommended. Instead of taking all 6 of your samples and running them on different lanes, or maybe you're even running this on one lane and this on one lane, that's creating false differences between how you're handling your control and treatment samples. A better way would be to take your 6 samples, when you have your libraries, split them evenly across your lanes. So sequence all of your samples at the same time. Don't ever treat your experiment and your control differently, except in your one experimental parameter. That's all you want to alter. Everything else should be the same. So that's, if you're talking to a sequencing facility, try to tell them that, like, don't put all my control in one lane and all my experimental on the other, because that'll have confounding results.
If all my samples can put in one lane, is that the best way to do that?
Yeah, that's fine. If you're not doing multiple lanes, that's fine. Just do the same library prep, just treat everything the same and then run it on the same lane. And if you're gonna buy 2 lanes of sequencing, just split your libraries evenly across both lanes. That's my recommendation. Was that your question too? Yeah, so these are people who are sequencing really, really, really deeply. This would be like, a lot of money for sequencing. But I think the principle holds. Like it's a basic principle. Just treat your samples equally and try. If you know there's going to be variation, like don't send all your controlled treatments to UT Austin to be sequenced, and then all your experimental treatments to Wisconsin. Like, don't do something like that. Yeah, so you could randomly assign your controls and treatments to lane 1 and 2. So you would just block out the lanes. So you wouldn't have to sequence if it wasn't feasible to sequence every sample on both lanes, just don't divide them between control and treatment, randomly assign them between the 2 lanes so that you've controlled for that. Does that make sense? Yeah, 46 in one, 46 in another. Yes, or block out like half my controls in this lane and half in this lane, and half my treatments in this lane, half my treatments in this lane. Oh yeah. So hold 80 samples, 46, not many, 46 other. Yeah. As long as you pay for the whole thing, yeah. yeah, it's your money. You can tell them how to do it. So yeah, I would definitely say a service facility should be aware of this, and hopefully they would be advising you to do that, not only like working with you to do it, but telling you needed to.
Really, what you would get is just more reads for 46 versus what you need.
Right. If you split like 80 and 12, then your 12 would be really, really, really deeply sequenced, and your 80 would not be. So yeah. Right. So I try to, yeah, depending on how many lanes you're going to buy, I try to put an even number of samples. Or if you have a low enough number of samples, you can do like this and run every sample on every lane. So once you've mixed all your libraries together, you can just put them on every lane. So that's how, yeah. There's a number of ways to do it. Just think about it from a statistical perspective instead of from a convenience perspective. Okay, so I've sold you completely on replicates now, right? Like, but there is actually, you have to balance that with read count, and there are some considerations with depth. The ENCODE Consortium, which was a big human sequencing NIH initiative. They also did a lot of model organism sequencing, such as the mouse data. They came out with these recommendations based on a lot of statistical analyses. If you're comparing two mRNA samples for differential expression, they recommended 30 million paired end reads per sample. However, if you were discovering novel elements, you need really precise quantification, really lowly expressed transcripts, 100 to 200 million paired end reads per sample. So if you have like some elusive little gene that you really want to study and it's very lowly expressed, you're probably going to have to sequence deeper than the average researcher who's just looking at transcripts that are pretty common. So that's something to be aware of. And that was from 2011. So I do feel like we can update that a little bit. We're now seeing the science is definitely pushing toward replicates and not depth. As low as 1 million reads can provide the same sequencing accuracy as greater than 30 million reads, was published in 2014. The caveat, again, this only applies to the 50% most highly expressed genes. So if you're really interested in lowly expressed genes, you're gonna have to sequence more deeply. And I think I already had this earlier. Beyond a depth of 10 million reads, for the most part, you're getting more bang for your buck with replicates and not with depth. So 5 to 10 million is usually where I recommend. If you can get at least that many per replicate, you're doing good. And yeah, be sure to look at what is currently being published in your community. I feel like this changes really fast. I mean, we're looking at recommendations from 2011, now already being turned over here by 2014. So I think the trend is definitely toward more replicates, but look at what's being published in your community. If you're working on microbes or yeast, you might need even fewer, even less depth, because there's fewer genes that you need to sample across. And also this can depend on what resources you already have. If you have a well-assembled and well-annotated genome, you can save money by using single ends and shorter reads. That can help you build up more replicates. However, if you're doing a de novo transcriptome assembly, you might want to look at doing longer reads, doing paired end reads. One of the things that I talked Jim into was you can produce one library for a sample and run it on both a MySeq and a HiSeq. So you can spend a little bit of money, generate your really long reads on your MySeq, use that for your de novo assembly, and then also run that same library on the HiSeq and get your depth. So there are some hybrid solutions here. You only have to pay one library prep fee because those are expensive. So you don't want to pay that more than once. So that can be a nice technique. Okay, and so of course you have to balance all of this with cost because none of us have an infinite amount of money. So I put a list of places that have publicly posted pricing. So for me, the first thing I do when I have a new project is get the budget out, get the experimental design figured out, and then go visit all these places and figure out how to balance library prep fee versus lanes versus instrument. I have worked with the UT Genomics Tour and the the University of Texas Austin Genomic Sequencing Service Corps. They're both really good at helping you with your experimental design, with figuring out And sometimes you have to be very careful when you're reading pricing lists. Sometimes there's like hidden charges for like QC, or like there's an extra $10 per sample handling fee. So sometimes it's worth contacting them and trying to figure out like those extra fees. But Science Exchange will also point you to lots of other places. All right. So do you guys have any questions about cost, read count, and replicates, and how to balance them? No? Vitaly was like the person who would start all the conversations, and now that he's gone, it's a very quiet class. Y'all should jump in there and let me know if you don't understand something.
So if you were reviewing papers, they pull their sample out of fatal law.
I would complain about it. But that being said, and this is so embarrassing, I have recently published papers with pool samples. And that's because when I was a postdoc, I was key personnel on a project, and the PIs just did their sequencing and sent me the sequences and said, here you go. And, you know, the scientific advisory board complained about it. I complained about it. Reviewers have complained about it. We ultimately published some of the data, but it was a lot more difficult. I mean, we did have reviewers ask for RT-PCR and kind of question some of the legitimacy of our results. And some of it we never, I ended up, the PI is still really mad at me. The last set of data I never even tried to publish because I didn't think I could get it through peer review. So that being said, I still think I see the papers come through sometimes. It kind of depends on the types of questions you're asking, and if you're more characterizing and generating preliminary data, then if you're saying, I'm 100% sure this gene is differentially expressed and we need to go after it as a target for this biological phenomenon.
The motivation.
Save money. So if it's a $250 library PEP fee and you're paying that, you're going to take all 8 of your biological replicates and pay once instead of pay $250 for each replicate. It's just super expensive to do the reps. Yeah. Yeah, like I feel like yeah, I've been tempted to just write that in a discussion. Like, I understand that this was statistically a terrible idea, but we did it because we're poor. Like, cut us a break.
Probably overly honest science.
Yeah, that would be another good Twitter. Yeah, overly honest.
I have a question about the number of reads. Because yesterday you told us basically you prefer like more than 10 million reads per sample, right? So if that 10 million reads means R1 plus R2, like forward sequencing, or
Yeah.
it's just
So reads are read pairs.
so
Yeah.
the total, or like total should be 20 million, right? Or just each.
It would be 20 million reads, 10 million read pairs. So, and this is a good point. Thinking about it statistically, if you take a single DNA fragment, even if you sequence it from each end, statistically you treat that like one sample. That's one ball that you've pulled out of the basket, right? So it's one thing that they could account for a gene. So you don't count it as, so I would still think about it in the scheme of the statistics, it's still, so 20 million reads or 10 million read pairs, right? So when you move from single end to paired end sequencing, you're getting more data, but that pair is still statistically only as good as a single end.
In this case, if we design an experiment, if we want to reach the coverage, let's say 20 reads R1 plus R2, and how many samples we put in each lane will we calculate?
Okay, so you're gonna get the same number of reads or read pairs from an instrument. You're just increasing the length or the amount of data you get if you get paired ends.
Yeah.
So if you run a high-seq lane and you're expecting 240 million reads, if you run it in single end mode, you're gonna get 240 million single ends. If you run it in paired end, you're gonna get 240 million paired reads. So you'll actually get 480 million individual reads, right? Because it's all about the cluster density on the slide. So it doesn't matter how long that piece of DNA that's connected to the slide is, it's just.
Firstly, generate, like how many reads each gene firstly generate for IC. and then you'll divide it by.
Yes, yes. You would take, like, UT Austin's great because they say we guarantee a minimum number of reads per lane. And so you can take that number and divide it by the number of libraries you're going to stick on that lane and get your number of reads per library. Yeah. Yeah.
So if you only have one biological resident, and is it acceptable for RT-PCR that we published, or?
I mean, probably not like Nature, but yeah. I do see those papers still come out, and especially if you have, I work with some PIs. It's like really, really, really expensive and difficult to get their one biological replicate, and that's all they're going to get. They get one shot at it, and then if you go in and do your differential gene expression analysis, find the 10 genes that you're most excited about, go in, verify with RT-PCR. Yeah. And if it's something that you can justify, like there's no way scientifically we could have generated more biological replicates in a feasible manner, then I think the reviewers will take that into account. If it's something where you're just lazy and you just didn't plant enough trees or plant enough plants in the ground, I think they're less likely to buy that. But okay. Right. Well, but you could have, if you knock it out and you have individual, like Fong is doing this with her soybeans. She's doing transient expression of the hairy roots. And even though she's using one vector and one line, she gets 3 plants and then looks at the variability between those 3. Yeah, the same one. Because it's still like going to be slightly different pots and slightly different places in the greenhouse. And I mean, there's going to be a little bit of variability that you can, it's going to give you more robust. You can see how you can measure that variability in the genes. So it's good. Yeah.
So this may be too simple of a question, but why so many reads same sample? I view it as you've got a sample of RNA, then you're going to read it 30 million times. Why do you need so many reads?
So there's multiple orders of magnitude of expression. So you have, let's say you have 25,000 genes. Some genes you're going to get hundreds of thousands of reads in sample A and hundreds of thousands of reads in sample B, and your stats are going to be super robust. That's like the upper echelon of highly expressed genes. You're also going to have lots of genes that you have like 10 reads in sample A and 15 reads in sample B, because they're just very low expression. And so to do statistics on those, you have to build up coverage. You just have to build up the amount of sequencing you're doing. So it's like this exponential curve of like just a very small handful of genes are super, super, super highly expressed, and they're eating up a lot of your sequencing power. And then you come back down, and as you're seeing those genes that are like mid to low expression, and you're still potentially very interested in those, you have to build up the coverage to get enough reads to do statistics on those.
So it's basically cover everything.
Right, right. It's because if every gene were like kind of evenly expressed, we wouldn't really need to do all of this. But because there's this huge variability in how some are, how much some are expressed versus others, you have to do the depth. Yeah. And I mean, you can have really important things like a transcription factor flips on. It's never super highly expressed, but it like causes this huge downstream cascade of other things. But if you're trying to find that one transcription factor, maybe it's like buried with only, you know, it's not as highly expressed as everything downstream, but you really need to identify that. You have to build up coverage at that lower expression level so you can see it in your samples. Does that make sense? And that's why they were saying with the depth, you have to increase the depth to see those lowly expressed genes. If you, with like 10 million reads per sample, you're really only going to see, from the literature I've read, the top 50% expression. So if you had 25,000 genes, that means maybe only like 10 to 12,000 of those genes would you actually be able to do statistics on. So you've still got this huge swath that are either not expressed in that tissue at all, which happens, or so lowly expressed you can't do statistics on them, even though you've generated 10 million reads. So that's why.
So, we're talking about the transcription factors and the genes that they are affecting. How are you able to like decouple the cause from the effects using that?
So you're not. that is not a powerful, like this is basically going to be correlation, not causation, right? Like we can tell you, it looks like we have differential expression in this gene, due to this experimental treatment. You would then have to go in and do a lot of follow-up lab work to prove whether or not that was causative or whether that was residual, right? Like maybe that's just an impact, like a downstream impact instead of an upfront causative impact. That's where you get into really fun functional genomics and knockouts, and yeah. So this is more you're characterizing the profile and you're able to see correlations, but yeah. Any other questions? All right, a little bit more about file formats that we're gonna see today. The SAM format stands for Sequence Alignment Map Format. And this does indeed store sequence alignments. So once you've aligned a read to a genome, you want to capture that information about where it aligned. So if a read maybe it ends up on chromosome 1 at 2 million base pairs, you might want to capture information about, are there gaps between the read and the reference? Are there SNPs? Are there structural variants? If you have paired reads, did those pairs end up in a logically consistent location next to each other, pointed the right direction? Or maybe one pair's on chromosome 1 and one pair's on chromosome 3. You're like, oh, well, that could be interesting. Is your read aligned to more than one place in the location in the genome? How high quality was this alignment? Are we very confident that this was the right location to put this read, or maybe a little sketchy? And basically all of this is stored in a tab delimited plain text file, or that's a SAM file. So the strengths of this format, it's very flexible, so you can store lots and lots of different types of information. It's pretty useful for operations on very, very large sequence data sets. Probably one of the reasons that it has become ubiquitous is it has really, really, really good documentation. So if you're a software developer and you need to be logically consistent with producing a SAM file that will interoperate with everybody else's software, there's documentation to tell you how to do it. And there is this great software package called SAM Tools that allows you to manipulate the SAM files. And we'll be seeing that this afternoon, or this morning. So here's the basic structure. There's a header, has these little @ symbols at the top. Those are basically kind of like comment lines. Gives you information about what software produced this file, what the reference sequence was, so a little bit about just what's to follow. And then the bottom part has each individual read has its own line with information about where it's mapped. So the header version information, so the SAM format specification does undergo updates occasionally, so it'll tell you what version of SAM this is, whether or not the file has been sorted, information about the reference that was used for mapping, any processing or filtering that was done gets added to the header, and the software version that was used to produce the file. Oh no, they're gonna be like millions and millions and millions of lines if you have millions and millions and millions of sequences. Oh yeah, the header can be tens of thousands of lines. Yeah, it actually ends up, so like the chestnut genome has 40,000 scaffolds. It puts a line for every scaffold to say, this is a list of all the scaffolds that we used for mapping. And so that's just like 40,000 lines right there. They get added to the header. These files are huge, especially if you have big data sets. So here's, and then once you get down to the read mapping, it's not super important to understand all of this, but I want you to be moderately familiar with it so that, again, if someone sends you a SAM file, you can open up and say, yes, indeed, that looks like a SAM file. So the actual reads, the first thing on the line is the name of the read. Then there's a flag that gives you some information about how well it was mapped. And then it tells you where it was mapped in the reference. So chromosome 1, chromosome 2, scaffold 262. And then the position on that scaffold or chromosome where it was mapped, and some information about quality. And from there, just a whole bunch of other information that primarily you as a human being are never going to use, but your software is going to use. So if you do a head, it will probably look, the header lines will look something like this. The actual lines will look something like this. So it can be a little bit intimidating to look at. But again, it's good to have like that visual confirmation of, yes, this looks like a SAM file. So this would be the read name mapped to the scaffold. at location 950,180 bases. And it gives you the actual read and the quality. And then a whole bunch of other codes that communicate to your software how well the read mapped. The sister format to SAM is called BAM, and it is a binary version of SAM. And the reason why we turn SAM files into BAM files is they get much, much, much smaller. So this is a compressed version. It is not human readable. So if you do a header or tail, you'll get something that looks like this. That's because it's been condensed way down in the super compressed version. But you can convert it back and forth between SAM and BAM with that SAM Tools software package. But you can save up to 75% of your file system space. So that's why we use it. There's also a new thing called CRAM. So now I have SAM, BAM, and CRAM. Not too many people are using this, but I've kind of started to see it a little bit, like some software packages have started to use it because it's basically an even smaller compressed version. So if you see it, don't be confused. It's basically just a different type of BAM. And you can see going from a SAM to a BAM file, you get a lot of file system savings, and then you get a little bit more CRAM depending on what flavor of CRAM you use. So SAM Tools is a software that you're going to use to manipulate these files. htslib.org is the place that you find documentation and information about SAM Tools. If you're going to be calling variants like snips, BCF Tools is going to be also your friend. And they have a whole set of formats for variant calling that are similar to SAM and BAM. And we won't get into those in this class, but I want you to know that they're there and they exist. All right, so that is all that I have. Do you guys have any questions? Yep, okay. Let's take a short break, come back in like 3 or 4 minutes, and we will get Gilly started on mapping. Sure. Okay, go that way. So they should be identical. So you barcode each of your samples, it's like those 6 pieces of pad, and then when it comes off the right, it's out what read and here you have, this is a bad experimental design. Because let's say you have some intrinsic problems, you can no longer take it because they would have let's say you had two circles. Okay, so let me tell you about this. So let's say you had 100 clients. 50% of them are transgenic. 50% of them are automatic. And you want to transgenic. You put all of your transgenic in greenhouse 1. You put all of your control in greenhouse 2. Greenhouse 2 breaks in the outdoors. So now you've got your transgenic plants, they've been consistently watered, and your control plants, and they've had this huge drought. Now, if you sequence them, you have no idea if the differences are due to the drought or due to the transgenic. To prevent that, what would you do? Yeah. So how would you fix that experimental design? Yes. So you want half of your control in greenhouse 1 and half of your treatment in greenhouse 1. And the other half of your control and the other half of your treatment in greenhouse 2. And that way, if something happens in greenhouse 2, it's only happened to some of your samples. And you still have temperature, right? So that's what we're doing here. If something goes wrong in these lanes, your sample is no longer comparable to everything else. If you do this, if lane 4 fails, it doesn't matter. Because lane 1, 2, 3, 5, 6, everything's been treated consistently. Does that make sense?
Yeah. Right now, I'm going to move back up and just like do new styles.
Right, but in the greenhouse example, if you have greenhouse 1 and greenhouse 2, just because these plants are in drought, if you have a randomized block design, you can account for that. You can still use the samples. You can take the drought out of your statistics. You know that someone can have something that's not okay. So that's why you do this. If you take a statistics class on experimental design, you'll learn a lot about blocking, for all of your treatments. All of your cycles are treated differently, and you don't have these failures. Okay. No problem.
We all ran out.
No, the 5 okay. Yeah. Yeah. Okay, so type in your and then
You should go outside for 3 minutes to get warm again.
So it should be exactly what you type in on the SSH.
What is it? Mail transfers?
Yeah.
So if your SSH I think you had
Okay.
So okay. If I were you, I would just use it. Okay. I would just use this. Just ignore vowels. If this works for you, you can use that. Yeah. Multi-PC report. Yeah. Just email me that. I actually had I can actually check you off the list because I've seen you transfer it. So, the homework is just to email me that file. Just attach it to the email. To the email and email. Yeah. Okay, so go to your email.
During such a waste of energy.
Hi, everyone. We'll start the next part. So what we are doing right now, we will map our reads to the genome, to the reference genome. We are using a peach genome, which is very similar to apricot. So we haven't have an apricot genome yet. That's why we're doing like a similar species mapping. And right now, let's get into the interactive section. Like, so go to the our website homepage, and here, you guys are all logged in, right? Okay, so copy this command to your terminal, and we get the interactive section. So the software we are using to map the RNA-seq reads to the genome is called STAR. It stands for STAR. It stands for Splice Transcripts Alignment to a Reference. So it's doing splicing. Some software it's not knowing like which part for the eukaryote organisms. They don't know where is the exon, where is the intron. So some software they don't doing the splicing. But this software, if we provide the annotation file to it, it will know where is the splicing site. So it can map the RNA sequence to the genome. Okay, so this lab has the STAR mapping has two parts. One is generate a genome index. The second part is the actual mapping. So first, let's copy the genome data from our folder. So let's go to RNA. So go to your folder. Go to and enter your RNA-seq directory. Go to analysis folder. Then we will make a directory to put your genome data into it. So make dirt genome directory. Yes. Should be go up inside the RNA-seq. Okay. Okay, then let's move this folder to the upper level. Slow.
So it was underneath the RNA-seq directory. So we accidentally created it down here, moved it back up.
I know, it's super slow.
The server slow. Hit Control-C and try typing again. Yeah. So whenever it hangs like that, I think it's actually a terminal sizing problem, but it's not that the server slow. It's just that.
Okay, so right now your genome directory is in the analysis folder. Everyone get that?
In RNA.
It's in the RNA.
It's in the RNAseq folder. And right now, let's copy the. Let's take a look at where our genome is. So go to Laster, Mendoza project, UT10 genome. So in this folder, we will have P-Persica FASTA file and P-Persica GFF file. So these are the files we want to copy into our own folder, our own genome directory. All right, so let's do CP. And copy the whole path. And the FASTA file. So copy the FASTA file into our RNA-seq genome directory.
Yeah, if you the control C and start over.
So we are copying this file into our genome directory.
So if you guys are having problems where you're trying to edit, like you're typing on the command line and you're trying to edit it and like it doesn't appear to be doing anything, type command resize and hit enter. That should help. We've been having this problem with the terminals doing weird things. But it's only if like you're trying to type and like you're hitting backspace and like nothing's happening and control C and then type resize and hit enter. And then your command line should work better.
And next, we will copy the GFF.
Does anyone know what a GFF file is?
It's an annotation file.
Okay. So I didn't teach this one, but it's an annotation file. It says where in the genome your genes are located. So it'll say gene 1 is on chromosome 1 from 111,000 bases to 111,300 bases. But actually, it'll have the gene, location of each exon, it can hold the UTRs, it can hold basically any part of a genome that you want to annotate, you stick that into a GFF file. You will also see GTF, which is very similar. So if you're looking for annotation, that's GFF or GTF are what you're looking for.
So right now, if you go into your genome directory, you will see our two files. It's in a folder. And you can also take a look at their size. So one is 220 meg. One is 96. If you like to see what GFF looks like, you can head the GFF.
Yeah, we're going to use ectoplastic later, but we're going to master the whole genome.
So in our GFF file, it has the chromosome name and its feature. So like the first one is a gene and its location is from 40,340 to 51,000. And its ID So what's the whole name of this gene? So this is what GFF looks like. Okay. And so right now we will create the genome index. In the genome directory folder, and go to our lab tool page, we will copy the whole bunch of command here. You can just copy this and paste in your terminal.
So it can help when you're doing this on your own to open a text editor and create the command in the text editor before you try to create it on the command line. Because that's a lot to try to do on the fly with the prompt. So I'll often try to do these things in a text editor, and then if you need to change something, you change it in the text editor and copy it back over to the command line. Because clearly, once you leave this class, you won't have all this conveniently laid out for you. Don't actually read the manual and figure out what every little flag does, and figure out all the flags you want to use, and all the little parameters. So
Okay.
you want to go through what each one is?
So copy all the things and hit enter. Then we'll let it run. And it takes about 10 minutes, and I'll go through like what's their mean. They run. So we are running star. And the first one is run mode. So we're doing generate genome index. So the mode is genome generate. And the second one, genome directory. So we put our index in the current directory. So it's a dot. And the third one, genome FASTA file. So that's our input FASTA file. So we follow the page FASTA file, genome FASTA. And the run thread is asking like how many threads, how many nodes you have in your computer. So we currently running in one node. If you have more, you can run more nodes. That will speed up your index generating. And the next one is GTF file. So here we put our GFF3 file here. So it will use this file to recognize which location is exon, which location is genes. And then the GTF tag exon parent transcripts. So this is specific for GFF3 annotation file. If you have a GTF file, it contains a different format. So this one is black. Its default is transcript ID, but in our GFF file, it's parent. So we change, like we are changing the parameter from transcript ID to parent. So that's what's in the GFF3, but not in the GTF. And the last one is we are specific by our read length. So this one, we put 94 because our rate is 50, and this should be read length -1. So we put 94, sorry, 49.
So it should be said, we don't have these flags memorized, and we've set them up for you guys as an experimental run. But the actual way you do this is every time you run it, you get out the software manual and you read it. And you have to go through and read every flag and be like, does that apply to my situation or not? How do I run this? So it's a lot of like, this looks a bit impenetrable when you just look at all these SJBB, what the heck is that? It literally, every time we start with a new software package, we go through and read the manual and read all of these different flags and figure out which ones we want to use. And they give you pretty good instructions. So don't just run this. When you have your own RNA-seq data, you will actually have to read the manual. And it's also really nice to read the paper. It tells you about the software package as well. So this is going to take about 10 minutes, and I was going to try to give you guys an overview of how does read mapping work exactly? Why are we indexing the genome? So many of you hopefully, I feel like at some point I'm going to be teaching, and there are people who have like never really like used physical text Brussels is mentioned on, so it's a history book, on page 82, page 166, and page 187. So if you're really interested in Brussels, you can go look up those 3 pages without reading the whole book. So that's what we're trying to do with indexing. We're taking the genome and we're looking at every word. So in the case of this, what did you say the default word size was? Like 31, 27?
The default word size.
I'd have to look what the default word size is.
You mean the KMAR or?
No, there's probably a default word size for STAR too. You probably can't even, you can probably adjust it. But anyway, so it breaks the genome up into these words, and then it makes a giant index, just like the index of a book, where it says the word ATCGGGCTATCG is found at chromosome 1 at location 150,000, on chromosome 13 at location 200,000. So it's creating this giant index. And then when you take an individual read, you find all of the words in the read, you go look it up in the index, and then that's where you head to in the genome to try to map that read. So instead of taking a read, like let's say your genome is a billion base pairs, instead of taking that read and trying to align it to all 1 billion locations, which would take forever, you have this shortcut where you look at all of the words in the read, and then you go try to find those words in your index, and then you only go to those locations in the genome to map the read. Does that make sense? Sort of? It's just much faster than if you tried to do it all against all. Yeah.
So what's the relationship with the word size you want for your genome?
So you have to use the same word size for the read and the genome, but you can set the word size, and that's an important question, right? So your word has to be an exact match for you to even go look in that region of the genome. So let's say your reads are, for example, Miriam's working on something that is quite a bit, the reference is quite a bit different from the organism we're sequencing. So the reads have more differences. There's indels, there's different bases that have mutated. So that matters when you're looking up exact words. So if she has an exact word length of 50, that 50 mer actually doesn't exist in the genome anymore because it changes, that read will never get matched. So you can shrink the word size and say, I only need exact matches of 10 bases or 20 bases to try to map the read. At that point, what you'll see is a very significant increase in computing time because your index is going to grow exponentially almost with the increase of the number of words that have to be stored. And then there's going to be more places in the genome where there are going to be exact words. So you need to go check all of those places, see if your read maps. So it's a trade-off, right? If you want fast, you go with, if it's fast and exact, big word sizes. If you want slow, that it can handle more noise, smaller word sizes. And this is conceptually the same as you're running BLAST. So we'll do BLAST tomorrow. And it's very much the exact same. Now most of these read mappers, STAR and Bowtie, they are not going to deal with anything below about 96 to 98% similarity between the reads and the genome. If you want to go down below that, you need a specialized mapper. We're also looking at STAMPy and GSNAP. They're significantly slower. Something like STAR is crazy fast. If you think about what it's actually doing, it is crazy fast. But it relies on most of the bases being so. And that's why we give it the location of the genes, because our reads are going to be split between exons. Our reads can cross an intron boundary. So the software is smart enough to create words that cross over the intron. That's why we have to give it, if we want it to be really accurate and efficient, you need to tell it where the genes are. It's also going to preferentially map to words inside of genes.
Words, let's say, just by scrolling to word and searches everywhere. Just the next position. Yeah. So, once it finds a word, scrolling along and finds something that's the same.
Yes. Yeah. So it looks at all the different words and then it tries to, yeah. And there's a variety of very smart algorithmic modifications that you can read about in the paper. There's like over 100 different read mappers, and they all have their like special little sauce that they use to speed things up and make it more accurate. But the general like conceptual how it works in general is the same. So de novo, producing the de novo assembly is its own separate crazy different graph, however lumps or nuts. But once you have your reference transcriptome and you have your reads, you map it back to the reference in the same way. If you have a reference transcriptome, you actually don't have to worry about introns. Because you didn't sequence the introns, they're not. So you can use a regular short read mapper instead of an RNA specific short read mapper. The STAR will work just as well. So for teaching STAR, you can use STAR, it would be great.
So is there like an option that you can know run your experiment?
That's a good question. I'd have to go back to the STAR manual. I don't think so. No, that's a very good question. She's been running STAR against a reference versus against an assembly. Like against a reference genome versus against an assembly, you have to use parameters of STAR. Yeah, I have to go back and look at the manual.
Sorry.
And that's like the secret is that like every time I run something, I revisit the manual, make sure I remember because I don't remember. I can't memorize. There's probably 100 flags for this program. I don't remember. Yeah, I didn't think you did. Yeah. So when you index, you don't give it a GFF. I remember for Bowtie, you had to create a fake GFF because it required a GFF. And I had like a script to do that. But for STAR, I think it's pretty clean. Top hat.
What prompted to switch?
So if you go to the Bowtie website, they're no longer maintaining it. They are recommending that everybody move over to HiSat. HiSat is a perfectly great program. We chose to teach STAR because it's super easy to install and it's super fast. I haven't done a head-to-head comparison of STAR versus HiSat. I think they both work great. And I'm not sure which one's faster. But for the Bowtie, if you go to the homepage, you will see that they're recommending STAR than HiSat, which is their new significantly faster and just they've also released StreamPy. I don't know. Some other things I haven't tested yet, but they've updated a lot of that. A couple weeks. So they call it the tuxedo package because they came out in the original ones are like puff links, bow tie, top hat, and now they have like ball gown, string tie. And then they had HiSat, which I was like, you've broken the whole HiSat doesn't fit in the naming scheme. But yeah, they've updated a lot. I'm sure there is. I can't, off the top of my head, I don't know. For blast, it's 11 for nucleotides, 5 or 6 for protein. But I think it's much, much, much bigger when you're doing short read mapping. You're starting with billions or trillions of reads. You can efficiently map them to sometimes billions of reference bases. You need a pretty big word to make the whole process. I can tell you off the top of my head what it is. Usually I just go with it. Like if I don't have a special problem that I think is outside the realm of normal, I just use the default parameters. If we have problems, if I map the reads and a whole bunch of reads don't map, then I may go in and start messing with those parameters. But if everything maps to the default,
So it's already done.
how many people are done? Okay, good.
So if you take a look in your genome directory, you see a bunch of files here. These are all the index this software generated. And we are going to use it to map our reads to the genome. So right now, let's go back to our analysis folder. And create a 04 star directory. So we'll put our mapping alignment here in this folder. And enter your star folder. The reads we are using will be the one we trimmed yesterday, because that one remove the low quality reads. So we are going to use the good quality reads to map to the genome. And in our website, still a long command that's copied in the terminal. And hit enter. Then it starts to run. So this command, I make it bigger. So we are still using star, but different options. So right now we are only put genome directory. So we specify where is our genome directory, and we file in. So we put our FASTQ file in here. For this library, we only have single end reads. If you have pair end reads, you are going to put two FASTQ file. So one should be R1 FASTQ, and the other should be the second one should be R2 FASTQ. So since we only have a single pair, we only put one FASTQ. And run thread is same as the one we generated the index, still run in the one node, and out file name prefix. So it will generate still a bunch of files, including the alignment, the log file. So we want these files all have the prefix of early flower red tiny. So mine is finished. Anyone has problems on this step? No? So if you take a look at this, we will have the same file and log outs, a bunch of log outs. We can take a look at the final log out. So if you cat early flower tiny log final out. It shows like how many reads get mapped and how many reads are not mapped, and it gives you like a percentage, like why the reads are not mapped because they're too short. And 5%, 5.72% of reads mapped to multiple loci. So if you want the mapping stats, you can read this to do your like analysis, like how this library map to the genome. All right. And the SAM file is actually our alignment file. So all our alignment are put in these files, and it's human readable, but it's not machine readable, because if we want to visualize our mapping, we have to convert our result to a software. It's called IGV, a genome viewer, and in this, it's not recognizing the SAM file. So we have to convert the SAM file to a BEM. The BEM is a binary file, which is much smaller, and it's easier to deal with the BEM file. So we are going to use SAM tools to convert the SAM file to a BEM file. So still in our page, and here, copy this command. Let's put in the terminal, let it run. So it's converting SAN to a band, and we call this sorted band because it will sort the alignment as the genome order. Because in our SAM file, it saves our alignment randomly. In our sorted band, it will sort based on the genome. So the alignment will show from the genome beginning to the end. All right, and right now, if you look at the file size, let's put ls-lh. We see our actual alignment is 45 back, and our sorted band is only 8.5, so it's much smaller. And we have one more thing to do. We need to create an index for the band file. So we will run this command. Let's copy it. And the central index will generate an index for the band. We will later use this band and band index to put it in the IGV to visualize our alignment. Right. You have any questions?
Indexing is not really like indexing. It's a location-based indexing instead of word-based indexing. What you're going to find with a lot of software packages in bioinformatics, there's an indexing step. And you can think of that as just a way that the software is going to be able to freely operate on the data and then make your downstream analysis really much faster. So this is very much a theme with like read mapping, with BLAST, with viewing files. Like there's a lot of times an indexing step, and it's just doing some pre-computation to make things faster. Ready to trade?
Mm-hmm.
Yeah. Is it time, or are you going to teach more? I can't remember where you're going to switch up.
Yeah, so next is IGV.
Okay.
You have to show them how to download.
Right. Okay, so it's really nice to look at your data. Right now, we keep shoving things into software and getting results out. I can't really see the data. And so at this point, it's really nice to actually just look at how the reads mapped to the genome. There's a number of different ways to do this. I really like IGV, which is Integrative Genome Viewer from the Broad Institute. However, it runs on your local computer. I can't make it run through the terminal. So that means we get to transfer files again. Yay. And we also have to install the IGV software. So if you search for Broad IGV, let me go back to this. It should pop right up as integrative. You say integrative or integrative? Integrative genomics viewer. I don't know. I feel like all these words that I like read so much more often than I actually say out loud. I'm like, turns out I don't actually know how to pronounce that. You can go to downloads. And you can try this quick launch, but I think it's just going to install the software anyway. So maybe more efficient to just download for Mac or Windows and get it installed. Actually, I don't know if the software is already installed on here, so we'll just go through the whole process. Okay, so I'm going to open it up and then double click and make sure it runs. It's gonna ask me, are you sure? Oh no. Okay, we may have to switch to my computer. So if you get a Java runtime environment error, hit up a red sticky. I don't actually know what to do about that, except you might have to reinstall Java, which might be a bit of a process. But I can switch over to my computer, and I'm not logged in. That's okay. So I'm not logged into Beacon on this terminal. This is just my terminal to my laptop. I'm gonna CD to my desktop. I'm gonna make a directory called IGB, just so it'll be like super easy for me to delete it after the class is over, which I have already done. So if I CD to IGB, so I've already got the files, but I'm gonna show you guys how to secure copy the files. So I would do secure copy, mstate1, login.beacon.nix.utk.edu, colon, so the colon always goes between the name of the server and the full path to your resource, the file you actually want. Okay, so the likelihood of me having typed this whole thing correctly seems very low, but we are going to give it a go. Oh, just changed, 427871. So this is gonna take, oh, it's speeding up. Okay, I said a minute and 10 seconds, but it's going much faster than that. So we have on the lesson plan the paths that you need to go get. So you need to go get the, is it cut off on anybody else's? Anyway, you should be able to copy it. So you need the genome. We have indexed the genome. You need the BAM file, and you need the index for the BAM file. So you should end up with 4 files. This ppersica.fa, this ppersica.fa.fai. You'll also want to get your GFF3 files so you can see where the genes are. The BAM file and the BAI file. Actually, you don't have to worry about. I think this one, IGB will generate this .fai. Yes, so you need the other 4. The only one you don't need is the .fai. Ignore that one. The IGB is going to create that for you on the fly. But try to transfer over the .fa, the .bam, the .bai, and the .gff3. And I think all 4 of those, the instructions are here. So I'll just do another one. Okay, so in this case, what did I do wrong? Anybody spot the problem? It says usage, like clearly you're not using this right. Oh, you can't see it? Should I try to make it bigger? I can make it bigger.
Hit the destination?
Yes, there was no destination listed. So sometimes the errors are a bit cryptic. They just yell at you and say, you're not doing it right. You're like, oh, but why? How? Give me more information. So in this case, this is not the greatest error. But if I add the dot, like copy it here where I am right now, it's going to work.
Is your local Windows?
Yes, this is local. This is not logged into
Okay,
Beacon.
okay.
This is me copying. So you, from a terminal on your computer, you're saying, go out to Beacon, grab this file, and pull it back down to my laptop. Yeah, on the secure copy command, if you add a -r, that means recursive. So if you wanted to copy a whole directory from Beacon, you could say scp-r, remote directory, local location, and it'll get that folder and everything in the folder. So the -r can be very useful. All right, everybody got the files? Getting there? Give you a couple more minutes. I guess I should make sure I have IGB installed on here too. I'm pretty sure I do. Yeah. Okay, so if you're having problems getting your 4 files, put up a red sticky. Otherwise, go make sure you have IGB, make sure that when you launch it, it runs and looks like this and you don't get a Java runtime error, which probably means you have to install Java. When you're ready, if you could put up a green sticky. I don't want to move forward until I have the majority of the class with me. Text. Or you have to reset your PIN. So you reset your phone. So Control C. Okay, so and just your password. Yes, just that all by itself. Yes, so now you have to create a new PIN. So you can use the same PIN you used to use. You just have to type it. Now I put it to okay, just try that. Or yeah, you can just try to log in. Okay, other end. All right, it's working. So now you can go back and talk about.
Thank you.
No problem. All right, I'm going to show you how to load these files into IGV. So the first thing is there may be some stuff up here, there may not, but we want to load our genome. And if you go to Genomes, load genome from file, you're going to go select your genome. So that's the P-Persica 298 version 2.0.FA. Open. You may get a pop-up that says it needs to be indexed. And I think you just click like continue or yes do that, and it'll index the file for you, and that might take a second. Did anyone get that? Yeah, did it work?
Yes, I just load that file.
Okay.
It didn't give me any message.
Oh, okay, perfect. So only the genome can you load from this Genomes tab. The other files we're going to load from this file load file. So first I'm going to select the GFF3. It says an index file cannot be located, an index file is recommended, and click go to create one now. Okay. I'm just gonna, actually I think it's gonna load it without an index file. I think I tested this earlier. It's not so big that it really needs an index file, but yeah, okay. So it's still loaded. Hopefully you have enough RAM on your computer. So here it's giving us for each chromosome, and it's kind of showing us where the genes are, but we can zoom in. So if you want to zoom in, you click and drag at this top bar. So I'm going to keep zooming in. So we're starting to see our genes. So we're on Prunus persica chromosome 1 from 14.3 million bases to 14.4 million bases. And we can see that these are the locations of our annotated genes. Now, if we want to load our, what we actually are interested in are RNA-seq data, right? Like, where are my reads? So I'm going to load from file and go get the BAM file. It says zoom in to see alignments. So we have so many reads, I can't even show them at this scale. So let's zoom in maybe on this one kind of big gene.
Just a second. What's the next file?
What's the what?
What's the next file after GFF file?
The BAM file?
Okay.
Oh, I'm getting an exclamation point. I don't know why. This worked earlier. Is anyone else actually seeing reads? sample sort.
I don't see alignment.
Yeah, something's wrong with our BAM file. I'm trying to figure out what it is. I think because you have to put the output before you put the input. Okay, so Luster, Medusa, Brush, ATN software, sample, sort, dash output, early flower, tiny. BAM, and then we'll put
You have to download the image version.
what's the index command?
It's index. IDEX. INDEX.
And then just the name of the BAM file.
This is the BAM file.
IDEX?
Or maybe the whole index.
Are you sure about that?
Well, we had used I mean, I just saw this. Yeah.
I don't know why it's doing that. It didn't used to, did it? I tested it last week and I could see things.
What's this?
I think those are S. I don't actually know what those are.
I tested on the large files.
And it worked fine.
I see pics.
Maybe there's just nothing in this region.
I didn't test this tiny file.
I don't see any reads. I don't know what's wrong.
Do you want to just
on the some file, some videos that should help with?
Can we just take a large file and dump it in?
Sure. Do you want to go find the large file and put it somewhere in the course directory? I don't have any large files.
It's no.
2982. EPP531-RNASeq-2017.
You get an error message when you load the DAX file. It asks you index file.
We are getting an error message, and we did not get an error message last week when we were testing the materials. So it might take me a while to figure out what's going on here. But yeah, this is a location where there should be a read, and there's no read. I don't know why. Do you want to put the .bam, the .bais for another one up in the course directory, and we'll copy it?
Yes.
Do you want to do that now?
You will put in the wiki.
Oh, I just put it in the project directory, the proj uttne.
It's already in
Oh,
the
okay.
directory.
Okay, I'll try it first to make sure it works. Next. I don't know why if we process the files the same way that would be a problem. I've probably never had that problem with IGV before.
It's in the analysis.
Forgot the colon. Oh, I forgot the file. Which one should I get? Any of them?
It's all in, so all the 6 files.
All right, so that file works. So when we process the whole library, for some reason that one shows up and our itty bitty tiny file does not show up. And so I'm not 100% sure if I'm just not going to the right places to see the reads for the tiny file. So it looks like for the tiny file, you just have to kind of hunt around to find your reads. When we load a big file, you can see lots of reads. So let me back out and see if we can see. Okay, so if I back out too far, we can't see the alignments anymore. But let's look at a single gene maybe, like right here. So that is a bad example. That gene might not be annotated very well. Let's look at this gene. If you drag and drop here, it'll move the whole window, whereas if you drag and drop up here, it'll zoom in. So let me try to get this gene here. Okay, so this is a good example of how RNA-seq reads are mapping to the genome. If this is your gene model and like the big blocks are exons and the little blocks are introns, you can see each of your individual reads is mapping between exons. So you see these bars, which means there's a gap. So it's mapping across the intron. So like here's another one. There is probably an unannotated exon right here. So this is why we like to use RNA-seq for annotation, right? Like there should be a little box right here indicating that this part is transcribed. But that could be the UTR, since there's like a sprinkling of reads in there. So if we were genome annotators, we might go in and try to figure out like why don't we have an annotation right here that says there's exon. So this exon looks very robust, right? Like this exon starts here and ends here, and indeed all of our maps, all of our reads map to that exact location, which makes sense. And same for this exon, that appears to be very robust, and these other exons. So this just gives you an idea of, are your reads mapping to genes? Is all of this making sense? You can also, if you zoom way in, let's zoom in on one exon, on just like a piece of an exon, we can actually see variants. So this is the peach reference genome, and we can see at this location, the peach reference is a T. Every single one of our reads is a G. So we know this is a SNP. This is some kind of difference between our apricot data and our peach data, our peach reference genome. So you can zoom in and out and see kind of how different your reads are. So this is our itty bitty sample at the top. This is a much bigger sample. So hopefully if you take that itty bitty sample and you scroll left and right, you should be able to find some reads. I think there are just so few reads that they're kind of hard to find. Yeah.
I just can load the file, file. When I load it, it says how index file is prepared for sample file.
So if you need to move over that .bai file, the index file is .bai. So when we did samtools index, it should have produced a file called .bai, and then put that in the same place as the .bam file.
I don't have that file. It's in that folder.
Okay, so here's another interesting one I'm just going to point out real quick. This one you can see that the bar is half red and half blue. So the reference is a T, and it's split between C's and T's. So it didn't put a T here because it's the same as the reference. So for every read where you don't see a C, it's actually a T, so it matches the reference. So we can see that would be a heterozygous SNP. So we did manage to get both alleles in this gene. Okay. Could you help me with the index file? Doesn't have a .bai. Is there everybody else? Was anyone else able to see? Okay. All right, that's good. And but it has to.
Yeah, right.
So this is sort of an underappreciated part of web informatics. People forget to look at their data, and you're going to find that the best QC is usually your eye, like just looking at it and understanding what it's doing, understanding if it's a mapping word, does this make sense? Maybe load up a few different samples, see how they compare to each other. If you have reads that are differentially expressed, or genes that are differentially expressed, you might want to go look in IGV and see if indeed you see a lot of reads from one sample and not a lot of reads from another sample. That would give you a good indication that your statistics are working. So I like this program for looking at data. There's also tablet. There's a couple others. Is everyone totally frustrated? Everyone's good over here. Everyone has. So this was the homework for today. It was to find at least, I think it was just find somewhere on IGV where you can see a read and see the gene and take a screenshot and email it to me to prove that you made it this far in the lesson. That's your homework. If you're an enrolled student, you're just here for fun. These are fun.
Okay,
we loaded up a much bigger set of reads.
So where are you at?
3 at 5,007,000. See if that's the same thing. So yeah, so mine is the same. If I zoom out, I've got one little tiny read too. So this, like, the top 2 boxes are little tiny sample file that we were using for the class so that everything would run fast. This is a much, much bigger sample file with 10 times as many reads. So your normal data would look like this. That's what we've been using for class data. So you're good. You're looking at the right thing.
Can now see the APCG like what you showed.
You have to zoom in. So if you zoom way out, there's too many bases to display. So if you zoom in, so that's the chromosomes. You're going up and down on the chromosome.
This
Yes, yeah,
okay.
correct. So here's some reads that
Okay.
you can see, and then if you keep clicking the plus, keep going. Oh, and there, your bases showed up.
Okay.
But you see how, like, when you're zoomed way out, you should all the bases.
This is a read.
That's a read.
Map to reference.
Map to the reference genome right there, and since it's all gray, it's not 100%.
Okay.
So if you put your mouse here and click and drag, yeah, you should be able to scroll down. So see, this read has 2 variants. So that read had a C and a T that differed from those bases in the reference.
These means gaps.
Nothing mapped there.
Okay.
This is a very small, sparse data set, so there's lots of gaps.
Okay.
But you would expect in RNA-seq, you don't expect to see a ton of reads aligning outside of genes, unless your genome was just very badly annotated. Or if sometimes if there's a lot of transposable elements that got activated and contaminated your RNA, you'll see weird things, but usually you expect it to follow along pretty closely with your genes. Oh, I think I might have caused IgV to, oh. Yes, so this is just one sample. You can also, like, just keep adding your samples in. So, well, this one I have our small set of files, our small file up here, and then our larger file down here. So I'm looking at 2 different lanes, like 2 different samples. So the very, very small file, and then this I loaded up was a bigger set of reads. So you could load up all, like, 6 samples and look at them all next to each other. After about 6, it gets too busy and you can't really see anything. But
Can we send you the screenshot?
yeah, just take a screenshot and email it to me. So yeah, this view is usually what will make me feel pretty comfortable that things are going well. I've got lots of reads that are just mapping on top of our genes and lots of gaps in between the genes. And you can scroll within these little boxes and see that, like, for example, here we have a lot of depth in this one region. And these little bars up here are depth. It gives you a little coverage lane.
It doesn't really matter because
Okay, so I think we have run out of class time officially. Sorry guys, we're moving a little more slowly than I had anticipated. That's okay. We will finish up tomorrow with HTC and move into, actually, raise your hand if you are interested in de novo transcription assembly. Raise your hand if you do not have a reference genome. So 1, 2, 3, 4-ish, maybe 3, 4, 5. Okay, so not too many of you. So if you guys are okay and have time to meet during the afternoon later this week, we could cut that out of tomorrow's and just go straight into everything.